session, so do not hesitate to ask all your questions, either on practical details or on global view on what we can do with single cell analysis.
Hervé, I did want to make sure that people knew that this was being recorded. So
okay, so this is recorded.
We'll record everything but the general discussion at the end.
Okay.
Thank you.
So now, so I want just to come back on the interest of doing single cell just before starting the talk. So we have worked for many years now on global transcriptomes, so mixing cells together and looking at the mean of the expression of all these cells. And the power of the single cell now is that we can really dissociate each cells and analyze each cell, so we can regroup cells that are similar and avoid to have just mean expressions. And nowadays, another interest is that we can also look where the cells are by looking at their expression using spatial transcriptomics. So I think these workshops are really to show you a bit the interest of all of this, the difficulties also to work on this type of information. But finally, it's very helpful to go on and to, for example, to discover new cell types, to work on cell-cell interactions, to better understand gene regulatory modules, gene regulatory networks at play in each group of cells, subpopulations that you are interested in. And I'm sure that we will find and describe many other applications that can be useful for genotype to phenotype studies. So now I will let the floor to Samuel. So Samuel, I let you share your screen. And meanwhile, I will present you. So Samuel Leiboff is an assistant professor at Oregon State University, and his lab is working on the developmental genetics of mice and other monocots. His team is interested in the mechanisms of tissue patterning and the use of single nucleus RNA sequencing to complement studies of anatomy, physiology, and genetics for this plant. So Samuel, the floor is yours.
Great, thank you very much. Thanks for the great invitation and great introduction there. I'm very excited to share some of the lessons that my lab has learned in trying to implement single nucleus sequencing for understanding development of maize leaves and other maize tissues. And so I'm not going to focus too much on the biological insights, but more of the technical lessons that we've learned. But I'm happy to talk about those too if you have interest. Because, me and all the folks in my lab were very interested in this question of how cells know what to do. And so we can think about that in a lot of ways, but my favorite way of thinking about this is a metaphor put forth by Waddington in 1957. Today we call this the Waddington landscape. But what Waddington proposed is that we can think of the development of cells as like a marble rolling down a hill or a blanket. And that we have a pluripotent initial state or a less differentiated state, and the cell will sort of go through its path of development into one of multiple final states. And so in plants, that might be something like becoming a guard cell, becoming a vascular cell, becoming parenchyma, becoming a mesophyll cell. What's really powerful about this metaphor is that even in 1957, what Waddington proposed is happening to shape the Waddington landscape is the activity of genes. And so this is the next figure in Waddington's chapter, where he proposed that we can think of genes as these little black pegs, and the way that they influence the landscape above that the cell is going to experience as these cables that stretch up to the fabric of development. And you can see that Waddington proposed that some genes might influence development on their own, but others might interact in a complicated network or interact in multiple, affect multiple steps in development, which is something that we know is true today. As a geneticist, I love this metaphor because it really puts the function of genes, the activity of genes, front and center. And we know that this is true, that if you remove some genes from an organism, then you can lose some cell fates. But I think that in this day and age of single cell sequencing, of very good transcript profiling, that this allows us to really think about development as a super dynamic process and capture these activities as they're happening. And so then what I mean by that is if we imagine this marble rolling down the hill, that its path will involve a bunch of intermediate states which are distinct. So immediately as that leaf primordia cell begins to differentiate, it enters into one of multiple states that will be distinct from the original. And the important decisions that that cell has to make, whether to become mesophyll or to join the vascular lineage, for example, those important decisions can't be predicted just by looking at the final tissue. And so then my group is very interested in trying to profile these things in action. And I think that plants and agricultural plants especially offer a really special model system for studying these types of activities because plant cells are additionally attached to each other by a rigid cell wall. And so that means that any of these important decisions that a plant cell is going through, any of these important states that it passes through, must be coordinated with their neighbors if there's going to be some sort of useful pattern that emerges. And that could be something as simple as dividing or not dividing, or that could be something like becoming a trichome or not becoming a trichome, that those decisions have to be coordinated if there is a final sort of pattern that's desired. And so then ultimately, how then do plants coordinate all of this activity of producing these sulfates to create these useful and functional organs with patterned tissues? And so then a system that my lab has been working on developing is understanding maize venation as one of these systems where we have the coordination of cells to produce useful tissues. And so in monocots, you're probably familiar with the idea that leaves have these parallel veins that run the length of the leaf. But if you look closely at a monocot leaf, for example, a maize leaf on the left, where we're visualizing high auxin levels using the DR5 RFP reporter, you can see that there are many parallel veins that are running the length of the leaf, but we also have some veins that are running perpendicular. And this is a class of vein called transverse veins. And those of you with really great eyes can also probably see that there are veins that are much brighter in the DR5 RFP, and there are some that are dimmer. And it turns out that there are many different types of veins or classifications of monocot veins. And I think that they're just a fantastic developmental system that demonstrate the sort of dynamic developmental traits that I was talking about before. And so if we look at the way that the veins are produced in the maize leaf, what we find is that at the beginning, it looks sort of like any old leaf, that the first vein to develop is the midvein in early primordial stage. And then as the leaf develops in maize and other monocots, we get the production of many primary lateral veins, which at least originally look like the midvein. In maize, we also get intermediate veins and small veins, which run parallel to the growth axis. And then finally, we get transverse veins, a special type of small vein type that runs perpendicular to the axis of growth. In the end, the pattern of the leaf in maize ends up producing a very high level of density, vascular density in what will be the photosynthetic blade, while there's relatively few veins in the sheath. And so we're super interested in the system and its implications for photosynthesis. And one of the reasons why it's such an exciting system for me is that we can sort of predictably capture these developmental time points just based on the staging of the leaf primordium. And what I mean by that is that we have these different classes of veins. In the end, we know that anatomically they'll look quite similar, and physiologically they'll perform similar functions for the leaf, but we can sort of catch them at different time points. And so then, because this is a single cell sequencing workshop, it's probably no surprise to you that this is an awesome sort of setup for looking at single cell developmental time points And this has been our goal for a few years. I'll tell you about the lessons that we've learned in trying to produce this type of data set. But the idea is that we can use these developmental time points to sample nuclei and characterize their expression as these different tissues are being developed and as they mature to really see like, okay, well, what's the deal with these small veins versus primary lateral veins? Are they really the same or not? And so when I started my lab in 2020, the very first way that we tried to address this question was actually using DropSeq. So the OG single cell sequencing platform with a few little variations. We tried to implement things as cheap as possible. And so we actually created a 3D printed syringe pump controller rather than buying real syringe pumps. And this was all great fun. And it worked quite well. We had the DropSeq chips printed by FlowGem. We could make emulsions and visualize, adding these barcoded beads with our cells or nuclei in real time. And my graduate student, Diana Ruggiero, thought that, this is kind of a low throughput experiment, but it was very fun. And it's something that we could, implement in the lab and sort of tinker around with. And it was very, very fun. But yes, it's a very low throughput technique, and I'm not sure that I would advise it except for some very fringe cases, or if it's set up already, then maybe it's great to sort of evaluate whether you're getting your cell type or not. But one of the roadblocks that we ran into immediately, even at this early stage, was that we were having a very hard time getting high protoplast yield for our specific cell type. So we were interested in catching these vascular cells as they're developing. But as you probably know, many vascular cells, when they are mature, are dead. And so it's very difficult to protoplast them. And so we started out looking at protoplasts. But what we found very quickly was that it was much easier to isolate nuclei. Nuclei can be isolated just by chopping in a buffer and filtering. So what I'm showing you here is just sort of the crudest method of getting nuclei. And so they're stained here with DAPI, which is identifying the chromatin. And so using fluorescence microscopy, we can see that, okay, just even at room temperature, chopping up some nuclei in a stabilizing buffer can yield many, many nuclei. And so then we work to sort of clean up this technique a little bit and make it a little bit more appropriate for getting high quality transcriptomes. And at first I thought that would be a very short effort, but actually is quite a long effort with a lot of trial and error. And so I'll skip over some of the trial and error and tell you about our best protocol so far. And so we really like the use of a per-call gradient for isolating our nuclei. And we find that it works very reliably for us, scaled up, scaled down, big tissues, individual organs. It seems to work quite well. And so if you follow the link on the slide, you're welcome to try our protocol as a Google Doc. But the idea is that we take that crude extract and rather than use it directly, we float it on top of a 25%, 75% per call gradient. And then in the end, at the interface of our 25, 75 per call gradient are the nuclei and any chloroplast. So unfortunately, the chloroplasts have to be removed through other means, use of detergent. But we don't find them to be a huge challenge for our preparations. And then as an example of the way that nuclei look after this type of preparation, you can see the figure in the middle where the nucleus is stained with DAPI. And then in red, you can see an example of one of those unfortunate little chloroplasts. and its autofluorescence. And so we're not the only folks who use this type of technique. If you want to see more, the Plant Cell Atlas recently had a single cell workshop that is recorded and available on their YouTube. And Dr. Joseph Swift, who was working at the Salk Institute, has a great video where he describes some of the detailed technical, or some of the technical details of this type of per-col gradient purification. But I will point out some of the things that we did learn along the way, although I don't want to bore you with the details of developing this protocol. So we find that the per-col gradient is great because it removes starch granules. And so depending on your tissue, especially if it is a photosynthetically active tissue, you might have a ton of starch compared to the number of nuclei. So on the left, here's a hemocytometer image where all of those shiny little objects are actually starch granules. And you can see that there's maybe a few nuclei in blue glowing there, but they're way outnumbered by the number of starch granules. And so these starch granules can clog microfluidics. They can probably break an emulsion. We never tried, but per-col gradient removes them very easily. We also learned that nuclei love clumping. And they're very easy to form aggregates, and that these aggregates will lead to multiplets in your final sequencing or clog microfluidics again. And so we always include bovine serum albumin, BSA, in our protocol, and we gently vortex our nuclei pellet to resuspend. That is quite difficult to resuspend a nucleus pellet otherwise. And at first, we weren't really that concerned with each individual preparation in checking its quality. But over time, we've learned that that's super important, and that between days, between people, between preparations, that your nuclei can look very different. And so we always try to look using a chromatin stain like DAPI at high magnification to see how nice our nuclei are looking. And the 10x Genomics representative says that we should look for crisp cookies, that your nuclei should look relatively round. The nucleolus should look like a chocolate chip in the center. And so we always try to look for that. Okay, so then that's the protocol of how to get nuclei. Then what do you do with them? So we've tried a few different techniques, and I'll tell you about what we think of them. So one of the techniques that I was most excited about after working with DropSeq for a while was to massively increase our throughput by combinatorial barcoding. And so if you haven't heard of techniques like this, combinatorial barcoding, split-seq, combinatorial indexing, the idea is that you fix the cell or nucleus and then perform in-nucleus ligation to create a random path that will assemble a unique barcode for many cells all at the same time. And the way that's done is in a 96-well plate, by splitting your cell suspension amongst that 96-well plate, pooling it together, splitting in a new 96-well plate that will add an additional unique barcode fragment, pooling and splitting and pooling and splitting. Sort of creating this barcode within the cell itself. And so in theory, this can produce many millions of unique barcodes from a high number of unique samples. Another advantage is that the cells and nuclei have to be fixed, and so that it allows you to preserve your tissue until you're ready to run the experiment rather than working with fresh material every time. But unfortunately for us, I know that Mark Lebolt's lab has had different outcomes, but unfortunately for us, we didn't have great success with this. And so what I'm showing you here is the final output of many months of work with one of these techniques, where in the end, we had a very low detection rate of about 5% of our input of, I think in this case, 100,000 nuclei. we only captured transcriptomes from about 6,000 of those. In the end, we had a very low number of transcripts per cell, 165, which came from only 87 genes per cell. So very low representation of the cellular transcriptome. And we also found that only about 7% of the reads generated in this library actually mapped to the transcriptome, and the majority of reads actually mapped to ribosomal genes. And so this was not appropriate for identifying marker genes in our analysis. And physically, this type of combinatorial barcoding involves a lot of repetitive pipetting. It's a very long technique. It is very appropriate for a robot, but kind of difficult. And so my student, Diana, said that it was complicated. She gave it 2 out of 5 stars overall. Unfortunately, although we're very excited about this sort of technique, we found that it wasn't usable for our purposes. I hear that recently there are other competing technologies that use combinatorial barcoding, but have different chemistries that might have better outcomes. But unfortunately, we haven't evaluated those, and we're a little bit scared given our past experience. So then, of course, we wanted to compare these results with the industry standard. So we also performed single-cell sequencing in a different experiment using 10x genomics. So we used a low throughput or LT kit, which is they no longer sell that. And maybe there's a reason why. So although we prepared 4,000 nuclei to do 4 reactions of 1,000 nuclei each, we detected over 7,000 nuclei in our final data set. So that suggests that there was sort of a problem with detection or capture or maybe free-floating RNA in our sample. I'm not sure, but that led to a nuclei detection rate over 100%. In the end, for the cells that we characterized, we saw just over 300 transcripts per cell and 300 genes per cell. So still a very, very low representation of the transcriptome. Although the 10x genomics process was very easy to run, and the proportion of reads that mapped to the transcriptome was 60%, so a very usable number of transcripts. And we did detect some of our expected marker genes, but unfortunately for this specific experiment, also wasn't the data we were looking for. And so Diana rated this 3 stars out of 5, that it was straightforward to run, but maybe there were some issues that were specific to the kit that we used. And so maybe we would need a different type of chemistry at a higher price point in order to make it work. I should also mention that the 10x genomics workflow requires a specialized instrument, which is quite expensive. Many institutions might have this instrument already, but my institution did not when I arrived, so it was quite a challenge to get one. But the final technology that I want to mention is one that we really like. And this technology involves cell suspension instant partitioning. That's the terminology for this type of technology. And the way it works is by using these special beads which interact with your nuclei or cells and then template a very uniform emulsion, even if you create the emulsion by vortexing. So there's no special microfluidics involved. There's no special well-based pipetting. The specialized technology is in these beads, which make a very uniform emulsion. And so this technology was offered by Fluent Biosciences when the data that I'm telling you about is from the technology as sold by Fluent Biosciences. They've recently been purchased by Illumina, and now their technology is available as an Illumina product. And so then the technology that I want to tell you about is PIP-seq. And so we love PIP-seq. It worked very well for us, very easy to use as well. And so we ran a series of reactions that, with 40,000 nuclei and detected 30,000 in the end. So we recaptured about 76% of our input nuclei, and these were from individual organs. And we detected over 1,000 transcripts per cell with close to 1,000 genes per cell. So much higher than the other technologies that I described to you. Still not, record setting, but we're working on trying to improve that. Our total percentage of reads mapped to the actual transcriptome was 74%. And we detected marker genes that we were looking for here in this analysis. Diana rated this 5 out of 5 stars, that the ease, simplicity, and speed of this technology really makes it seem like this could be the accessible future of single cell. And one of the reasons why we say that is that this technology scales up to very high numbers of nuclei, and it also scales down to very low numbers of nuclei sort of equally well. And so for our goals of doing these individual organ time points, this suits us very well, whereas some of the other technologies don't scale down very well at all, as we learned. Okay, and so then those are some very technical lessons that we learned. And I'd love to stop there and thank all the folks involved, and of course the support by generous funding agencies and opportunities. And I'd be happy to take any questions that folks have at this point.
Thank you, Samuel. There are some questions in the chat. You can access the questions?
Unfortunately, I cannot.
Okay, so I will read the questions. So one question is, have you tested your Percol gradient methods with frozen ground tissues?
Oh, that's a great question. So how we tested our gradient with frozen ground tissue? Yes, we have. Unfortunately, at this point, we haven't optimized the use of frozen ground tissue. We find that there's a lot of debris in our extract if we use frozen ground tissue. And there's at least a 10-fold drop in the yield of nuclei. So because we're trying to characterize these very small organs, we've put that on ice for now. But we've definitely seen nuclei. They seem reasonable quality, but the yield is much lower than our target. So hopefully that addresses your question.
You should.
Okay, so yeah.
Samuel, you should be a co-host. Can you see the rest of the questions?
No, right now I see everything as a participant.
Okay, no. I have one question. How do you explain the differences between the three technologies? It's because for parts you are fixing the nuclei, or do you have any idea of what could be the problem?
That's a great question. So along with Bruno Guillotin from Ken Birnbaum's lab at NYU, around the same time we were trying to troubleshoot with Parse Biosciences technical representatives. And he was experiencing the same problems in Arabidopsis and in sorghum and in maize. What we sort of settled on is that the Parse Biosciences technology uses a random hexamer as one of its first RT steps. And so unfortunately, the way that they've designed their chemistry, it's necessary to use that random hexamer. And so I think that for us and the way that we were preparing our nuclei, that we just had too much contamination of ribosomes. And maybe there's additional steps downstream that could remove those ribosomes, but that compared to these other technologies that seemed a little more promising, that we just couldn't optimize it.
Okay, thank you. We will move on to the next talk, and we can answer the other question, like who is providing the PIP-seq, so it's Illumina now. But anyway, we can come back at the end of the talks. Thank you very much, Samuel.
Great,
So
thank you.
I let Adrien starting to share his presentation. I will just present Adrien. So Adrien is working in France. He has done his PhD in my lab, working on pig cells, embryonic cells, and he's now working on human microglia in another lab in Paris. He has some expertise in single-cell RNA-seq, single-cell multi-omic sequencing, and spatial transcriptomics. And he's using this type of information to study cellular heterogeneity, gene regulatory networks, and spatial organization of gene expression in different models, mostly in mammals. So I let you start, Adrien.
Adrien, you are muted right now.
You should be able to unmute now.
Yes, thank
Yes.
you. So as I say, I will present from my view of an bioinformaticians the problematic from different data set and try to choose which method suits the best to your biological questions. And what I have done is I've been working in single cell RNA-seq in microglia, in pig embryo and stem cells. And I also perform multiomics and spatial RNA-seq with stem cell and melanoma. And I will firstly present the quality, the control and the pre-analysis on what each cell type can affect those parameters. And then I will present the downstream analysis possibility offered by the RNA-seq, the multiomics, and the spatial transcriptomics. So the first step of quality control is the filtering for quality metrics with the filter of mitochondrial DNA transcripts, the number of expressed gene and the number of transcripts. And those parameters can be affected by the cell activity. For example, in the pig embryo, we got much more percent of mitochondrial transcripts than in the mice brain cells. So then the next step is the removal of doublets. And the doublet can be affected by the cell property and the dissociation method you have used, but it also been affected by the technology like InDrops, 10x or SmartSex. And for example, you can see here the data on the mice brain, and you can see that this big population of microglia and astrocytes have some difficulty to dissociate and have a high percentage of cells that have been clustered together that share a marker of microglia and astrocytes. And if we take back to the embryo, the number of doublets was really lower. Then the next step is the normalization and the gene selection. So there is two main normalization method used, which is the log normalization by SWAT and the SCT transform. And here the SCT transfer is mainly required if you use spatial transcriptomics, as a lot of package require this type of normalization to perform their analysis. And for the gene selection, it will depend on the diversity of the cell you will have. For example, if you have an atlas of neural cells, you will need more gene than if you selected a specific population, for example, with FACS, and you only look to subpopulation of astrocytes, for example. And to visualize this noise effect, you can compare here two datasets of brain microglia. One with an indrops, which is similar to the 10x 3' transcriptomics. So in the right, you have the 10x flex, which is an 10x method with selected genes. So in the selected gene, you will get less genes that will apport a lot of noise. So you can see here for this population of cells that will be more separated and more easily clustered with less lines in the right between the left methods. And another important things to have in head is that single cell is bound to give proportion because all cells doesn't have the same survival rate through the experiment. As for example, trophectoderm cell doesn't have or nuclei isolation when we perform the multiome than in the classic RNA-seq. So we have less cells in this experiment. And it can also be induced biases if you perform tissue section. For example, in our melanoma sample, we have done different tissue section and it have been affect the proportion of cells, despite the fact as we didn't see the expand of the melanoma in the skin. And the next step is clusterization. And the clusterization parameter you will use will depend on the level of subpopulation you want to describe. And it also depends on the cell annotation you will use. And to help you, you can use the package clustry that will indicate you how the cell will be divide in the different cluster depending of the resolution you use. And now I will present the downstream possibility that single cell can offer you. So for the single cell RNA-seq analysis, you can of course do differential expression between your cluster, and you can perform functional enrichment with diverse methods such as IU cells, GSVA or GSEA. And also SCPA is a good package for pathway analysis between condition or pseudotime because it has been developed to specifically two single cell methods. You can also analyze interaction between population with packages such as CellChat, NicheNet, or Cellphone. And you can see on the right here the cluster of hypoblasts that send the ligand to the trophectoderm and the epiblast here. And then you can also perform a gene regulatory network. So the package the mostly used is Scenic that will give you Regulon, which is the composition of transcription factor with co-expressed gene with a common motif that have been associated to this transcription factor. And here you can see all the regulons that have been identified, and you can then infer the regulon activity between all your cell type or between pseudotime. And for the multiomics, you will get RNA-seq information and attack-seq information for the same cells. So the first question you may ask is, which benefit do I have to have two information on the same cell? So for the attack-seq signal, you can see that the attack-seq is greatly similar between each of our subpopulation in the different population of here it's the embryo. So it's quite homogeneous between trophectoderm, epiblast, and hypoblast. But if you look at the RNA-seq information, we will start to see subpopulation more often, and you have more effect of batch effect of the different sampling you've done. So there is more diversity in the RNA-seq information than in the attack information. But sometimes you can also see example where the both information can be quite precious. For example, in the orange cluster, you can see that it belongs to the blue population for the attack-seq information, but it have a very different RNA-seq information. So with combining this two information, we can understand that this population is originate from the blue clusters. So with both this information, you can see here for the example gen gate A6 that you have here the attack-seq signal between the four different clusters. You can, from this, identify a peak of accessibility on the signal. You can also have the RNA expression between the different clusters. And if you combine both information, you can link the peak accessibility to the expression of the gene. And you can see here that there is a lot of peak that are really necessary to the expression of the gene, and that they are specific to this cell population. You can also do the difference in accessibility in promoter region, but here we can see between the different population of trophectoderm, epiblast, or hypoblast. And you can also see the association of accessibility of regulatory region with gene expression. And you can see that it clusters population the gene in a different category. You have a preparation for the implantation for the trophectoderm cell, for example, which is something we accept. And we can see here that the hypoblast is also preparing to give the endoderm cells. And one of the best benefits is to have a better gene regulatory network, as you can have two information. You have the transcription factor expression with the gene expression, but you can also look for a motif in the peak that have been linked to the gene expression. So here you have in the blue to red gradient the level of gene expression for the regulon, and with the size of the dot, you have the accessibility of the regulon. So the wider the spot here, the more open the regulon will be. And you can see mainly that here you have the activator. So more the region is open, more the gene is expressed. And otherwise, you got more the gene, more the transcription factor is expressed, less the gene is expressed, and less the chromatin is open. And if you combine all this information, we have been discover new transcription factors that regulate population. You got the expression of the transcription factor. You can see that the motif is enriched in the same population. You can find on the regulon is accessible and expressed in the same population. And one thing that works pretty well is the footprint of motif. As you can see, you can infer the presence of the transcription factor in your different cluster. And here you can see for the nanog motif that here you have in green the epiblast population, and you can see that the transcription factor is probably still bind to the DNA in the cell from those clusters, which is something that we expect. But you can do motif enrichment between your cluster, but it's difficult to know if an motif, if a particular peak can be linked to a specific motif. Because of two difficulty is the motif similarity between the different motifs from the same family. And in a peak that you have here, for example, you have multiple motifs that have been identified with pretty good score. So it's difficult to know which motif can be bind to this specific place. And one slide for the spatial transcriptomics. It has been much more challenging experiment, as we have more than 50% of our slide that will not be analyzable. And we have much more less resolution, as you know that we have multiple cells from one spot. And it can be difficult to annotate the spot. For example, here you have an immune response, but all the different type of immune cells are in the same spot. So it's difficult to see which cell will be interact in each other. And you can also have locality area. For example, if your sample will not attach correctly to the probe. And to conclude, depending on your question, if you want to go more in the gene regulatory network, the multiomics will be the best method. But if you want to go more to cell the interaction, it might be best to do spatial RNA-seq. But if you only want to see the subpopulation and functional, maybe single cell RNA-seq will be just an best option. So thank you for listening, and I'm now open for question.
Thank you, Adrien. I will open the chat. So there is a first question on the bias due to survivability. So how we induce this bias by doing the experiments. Do we have a clear sense of cell types which are lost in these experiments, and are these numbers reported in the publications?
We didn't accept that much because when we, for example, in the trophectoderm population, when we do the single cell analysis, we have the same proportion of hypoblast and trophectoderm. And we know that and when we do the single nuclei, it was very different. And it may be the nuclei isolation because all cell doesn't have the same acceptation from nuclei isolation based on their morphological property.
But to complete, I'm not sure we know exactly in the publications if people are describing these problems. Didn't see it so much.
Yeah, not so much.
Adrian, can I ask a question? I don't think the host can type into the Q&A box. So we're kind of handicapped in the question and answer part. But I was very interested to see that you were able to do transcription factor footprinting with the single cell ATAC-seq data. I thought that needed a very high depth. Did you find that you didn't need a very high depth, or maybe it's across all cells? Or I was wondering how you did that analysis.
In fact, the depth was sufficient, and it really worked fine for most of the motifs that have been enriched in the different population. And it's also quite surprising, like as you know, Nanook didn't accept to bind a long time. So normally it's not a motif that is good for footprinting. So it was surprisingly good.
Okay. Is that in your 2025 paper? That looks like.
Yeah, it will be. We are prepared. I'm late. I'm sorry. It's not Adrian's fault. It's mine.
Okay. No.
But we will submit soon. The point I think is also that the more cell you have in your cluster, your population, the best you can detect this information.
Sure.
It's not really the sequencing depth, but it's probably more the cell events, the number of cell events you have.
Which is correlated with depth of sequencing data you have, right?
In some way.
Yeah.
Yeah.
Very good. Thank you.
I have one more question, maybe for Adrian. So you mentioned that ATAC-seq data do not give you the same level of information to cluster your cells compared to RNA-seq, and it was very obvious from the two UMaps you show us. My question is about, the saturation capturing chromatin accessibility fragments and transcripts. Do we have basically the same kind of coverage of the two information with those two technologies? So in other words, does the bias between ATAC-seq and RNA-seq, is it a technological or is it really a biological significance?
Good questions. And in my opinion, it's more biological effect as from the different data analysis, but
yeah, technically it's a bit the same because when you do the ATAC-seq, you have like 20 mil captures over, I think in our experiments we have 100,000 peaks, and in one cell you will get like 15,000 insertions, something like this. And when you look at the genes, you have around 50,000 genes annotated, and you will get 5,000 genes in each cell. So finally, you just have a small representation of all the information, but it's more or less the same for both type.
Yeah.
So that's why you need more cells to really get all the information for one specific population.
Yeah, I agree with that. I mean, 10x reported maybe a 30% capture of the transcripts with their newest kit. And for some data we have basically we can support this number. I was wondering if they have any kind of assessment of the coverage of ataxic fragment release from one single cell or nucleus in this case. So we can have basically a similar estimation. But it's not easy. Thank you.
So Mark, I think we can follow up.
Yeah, absolutely. So thank you very much, Adrian. And I'm pleased to introduce our last speaker for today. In this case, Sunil Raju. So Sunil got his PhD at the University of Nebraska-Lincoln and then moved as a postdoctoral associate at Michigan State University, then in New York University. And so more recently, he started as an assistant professor position of plant resilience in the Department of Botany and Plant Science at the University of California, Riverside. So in his lab, Sunil is working on understanding the evolution of cell type-specific stress response, including, of course, the genes driving those responses across different crop species. And so the idea is for him to use also the wild relative of those species, of those crops, as model systems. And so in addition to the research interest, he's also dedicated to mentoring the next generation of scientists, so different type of organizations, such as the plant postdocs and the plant grad that provide to those researchers unique resources for career development. Saying that, Sunil, now up to you. Thanks.
Okay, thanks for the wonderful introduction, Mark, and thanks for the invitation to come and share some of my research here. I guess the first two presentations talked more about the technical details of single-cell sequencing technologies. So I'll talk a little bit about some of the applications that we can use and biological information we can get from these single-cell sequencing technologies. So we started with the question, how can we understand cell type specific expression, and how can that inform us to understand better about the functions of genes, particularly in maize? So plants have this unique ability where they produce different meristems, and these meristems are important for producing different organs, such as the shoot apical meristem produces different organs such as stem, leaves, and flowers. And the root apical meristem is important for producing different types of roots, such as the primary root and the lateral roots. But when you look at these organs, they are generally made of these three important cell types or tissue types. The vascular tissue, which is the xylem and the phloem, important for movement of water and photosynthesis through the plant. And then we have this ground tissue, which in the leaf is important for photosynthesis and other purposes. And the final is the outermost layer, or the dermal tissue type, which is the outermost covering of the plants. So plants are reusing similar cell types to produce organs, but these organs are performing distinct functions. So the question that I had was, how does homologous cell types in different organs lead to distinct functions? So I used maize root system to try and decipher this, because maize produces different types of roots. So maize first produces these primary roots, the thick root, which is the first to emerge, and it's important for water uptake and transport. And then maize also produces these seminal roots, which are much more shallower, and they are important for water uptake, but along with that, they're also important for nitrogen and phosphorus uptake, particularly from the top layers of the soil. And then we also see these tiny roots in both the primary and the seminal roots called the lateral roots. So these lateral roots just grow for a short length, and they cease to grow after that. But they are much more important for water uptake, particularly in the later stages of the plant. And the water uptaken by those lateral roots are transported by primary and the seminal roots. And we also have crown roots in maize plants. These are roots below the surface of the soil. These roots are important for water uptake, but along with that, they are also important for anchorage. And these are unique to grasses. So with these different types of roots, now you can see that they perform overlapping functions, and still some of them also have unique functions. But when we look at their anatomy, they look very similar. So here I'm showing anatomy of the primary, seminal, and the crown roots. You see that they're formed of a central cylinder of steel, followed by pericycle, and the endodermis, which is the layer that modulates the movement of water and solutes into the steel, and then multiple layers of cortex, and then epidermis. And apart from lower number of cortical cell layers in the seminal root, they anatomically look similar. Going back to my initial question, how do similar cell types perform distinct functions in distinct organs? So for this, we used single cell sequencing in these 5 different types of root tips. So we took the root tips, we isolated protoplast, which is basically removing the cell walls, and then the cells with the cell membranes are in a solution. And then we use this 10x genomics library preparation method. So basically what it is doing is it's a microfluidics based system where each of these protoplast cells are combined with unique barcodes here. And in each of these droplets, you can see that we have one unique barcode and then a protoplast cell, so that all the transcripts that are coming from that one cell will have that unique barcode. And then we can sequence them. But at this point, we only know that all the transcripts are coming from one particular cell, but we don't know what cell it is or what cell type it is. So we use computational methods to look at that. So this is an example of a 2-dimensional projection where each dot is a cell and they're clustered together based on their expression similarities. So 2 cells in blue have much more similar transcription profile compared to another cell in orange or in green. So with that, I did this single cell transcriptomics in these 5 different roots and then combined them together into an integrated object. So here you can see that now again, I'm seeing good clustering here. And in this cluster, you see that there are cells from all 5 types of roots. So this suggests that these cells are clustered together based on their transcriptional similarity, irrespective of whether they come from the primary root or the seminal root. But again, at this stage, we know that they are clustering separately. That means they have some unique transcriptional profile compared to all other clusters here. And then we can annotate this into different cell types. So for that, we use known classical markers that we know their expression domains in, and then we use them to annotate the clusters. So for example, here you see that these 3 genes that we know are expressed in the endodermis in maize are only expressed in cluster 4. And instead of naming it as cluster 4, now we can change and say that this is an endodermis cluster. And similarly, we can do for epidermis and root cap and other cell types. So this way, instead of giving them cluster number, now we know that the cluster is either columella cells or root cap cells. And this way we can annotate the entire UMAP and say, now we know what cell type they belong to. So in this cluster, now we call this as phloem cluster. And you remember that this cluster had cells from all 5 types of roots. So we can go back and look at what is conserved in the phloem of all of these root types, or we can also look at what is specialized in one of the specific roots in the phloem cells compared to all other root types. So we also do validate these to make sure that these cell type specific markers that we see from our single cell sequencing are also true. So for example, this gene is only expressed in the xylem here. So the red means that higher expression of the gene. And then we can validate that by looking whether this gene is present in the xylem. So here again, you see the magenta color here in the xylem cells. And similarly, we can do that for other cell types. This is the gene that's expressed only in the endodermis based on our sequencing data. And then we can also confirm that it's only expressed in the endodermis in the root using fluorescent in situ hybridization. So from this, we can know that there are genes that are expressed in specific cell types. But how can we understand about function of a gene by looking at where they are expressed? So we looked at genes that are expressed differentially in a cell type specific manner. And one of the examples is the grassy tillers one, which is a classical maize domestication gene. And you see that, again, red here means gene is expressed in these cell types. So grassy tillers one is expressed in the meristematic region in both the primary and the seminal root, and also in some cells in the pericycle and in the xylem and phloem here. But when I looked at the lateral roots, I see that the expression pattern has changed now, wherein I see that it's expressed in almost all of the cell types at much higher level compared to the primary and the seminal root. So this suggests that there is change in both the abundance as well as in the cell type specification for this Gracitilars1. And again, we validated this in the lab using in situ hybridization. And I see that the expression, again, in magenta color here, is higher in the steel and in some pericycle cells in the primary root. And when I look at the lateral root, I see much more expression in all the cell types that corresponds well with our single cell sequencing data. But this still doesn't tell us about the function of these genes or how it is related to differences in that cell type specific expression. So for this, we use the mutants of Gracitilars1. So this is showing a wild type. And in wild type, Gracitilars1 is expressed in the auxiliary bud, which actually suppresses the tillering. And when you have a mutant of Gracitilars1, where Gracitilars1 is not expressed in the auxiliary buds now, we see the tillers formation. So we see a more bushy plant, a Gracitilar1 mutant. So this phenotype is well characterized in the shoot, but what happens in the root is not well known. And just to reorient you, so I took these root tips here for the primary root, and then look at the Gracitilars1 expression. It's expressed in the meristem and in some pericycle cells here. And then I took this the lateral roots from the primary, and then we looked at the expression pattern, and we see that it's expressed across all the cell types in the lateral roots. And when we look at the root architecture in the Gracitilar1 mutants, now we see that the lateral roots here are much longer, and we quantified this, and we see that there is increase in the length of the lateral root. So this suggests that Gracitilar1 is a growth suppressor. When it's present in higher quantities in the lateral roots, it actually suppresses the growth. Whereas in the mutants, when Gracitilar1 is absent, we see longer lateral roots. So through this, we have shown a new root function for Gracitilar1, which is already a well-characterized gene, suggesting at the power of single-cell sequencing technologies. But not just looking across two different organs within a plant, but we can also look at cellular transcriptome across species. So this is work from a colleague of mine, Bruno, where we looked at different root transcriptomes at the single cell level in maize, sorghum, and Citaria. And then we integrated this into an object where we can now start to understand about the functions, about the evolution of individual cell types. So with this data, what Bruno did is to look at how the cell types evolve across different species and how this could be related to the evolution of novel traits. So for example, he shows that among different cell types, epidermis across sorghum, Citaria, and maize seem to be more conserved in its expression pattern, whereas steel shows some divergence. And we saw that columella cells are the ones that showed the highest divergence in gene expression pattern across these three species. And when he looked closely, we see that a cell expression module that was present in sorghum for mucilage gene expression that has been co-opted in the columella cells of maize for production of mucilage. So this suggests that gene expression modules from a different cell type can be co-opted in a new cell type for the evolution of novel traits across species. So this again suggests that using single cell sequencing technologies, we can start to understand how novel traits evolve across species and how there is a lot of co-option of already existing gene expression modules to get into novel traits in a different species. So all this data set that I showed you here, we usually use the 10x Genomics library, 10x Genomics platform to generate libraries. But when we think of expanding single cell sequencing into more comparing stress experiments or across time course experiments, 10x Genomics library methods become challenging, both because they are expensive as well as some other limitations. So we, in Ken Birnbaum's lab at New York University, explore different technologies for the single cell sequencing and see which ones could help us in understanding this cell type specific expression patterns better when we have many more samples and when we have many more time points to sample. So with that, I'll talk a little bit about some of the technologies that we have explored in Ken Birnbaum's lab. So first is the 10x Genomics, which uses a microfluidics-based system that I described earlier, where the microfluidics is set up in such a way that one cell or nuclei here is emulsified in a droplet with unique barcodes, and all the transcripts that are coming from that cell will bear that barcode, and we can identify that these are all coming from the same cell. And the other technology that we tried in the lab is the single cell combinatorial indexing through scale biosciences. So here, as Sam was initially describing the parse technique, it's similar to that, where we have this 96 well plate, and we do this combinatorial indexing in multiple plates so that we get unique barcodes for these individual samples. So we can multiplex 96 samples or even more than that in the single plate and sequence them together through this combinatorial indexing. And we also tried this other micro well based technique through BD Rhapsody, where you inject your cells and beads through this into this micro well, and then have all the reactions happen in the same micro well. So I will describe a little bit about the scale biosciences and the BD Rhapsody, but I also want to mention that in our lab, both Bruno and Graham, they have worked on parse biosciences and fluent biosciences a little bit. So I'll talk a little bit about them in the later slides. So in the scale biosciences, as Sam was mentioning, one of the advantages similar to parse is that the ability to fix and freeze the samples. So this allows us to sample across multiple time points or across stress samples. And these fixed nuclei or cells are stable for up to 12 months and two freeze-thaw cycles. So once we have these fixed and frozen nuclei or cells, we can go through these indexing protocol, where first we use this index reverse transcription, where we have around 5,000 to 10,000 cells per well. And then we have this index ligation step. And after this ligation step, again, we can freeze these samples for up to 12 months, or we can go to this final index PCR stage. And in this stage, the input per well is around 1,600 cells. So in total from this 96 well plate, we can sequence up to 125,000 cells, and the output from each well would be around 1,200 or 1,300 cells. So from our data from Graham and Bruno, we see that when we used protoplast cells, we had 7,000 to 9,000 UMIs per cell. And when we used nuclei, we got 1,000 to 1,500 UMIs per nuclei. And then with the microwell-based BD Rhapsody system, this requires instrumentation. So we need to buy this HT Express cartridge system, which is around $15,000. And then the BD Rhapsody scanner, which I think is around $50,000. So in this system, what we do is we collect the cells or nuclei, and then we inject them into these flow cells so that each of these cells or nuclei go and fall into these microwells. And then we load them with these barcodes so that each of these microwell have one barcode and a cell. And then we can lyse the cell and then get all the transcript into the bead. And then we can retrieve the bead and then synthesize cDNA and then go through the library preparation. So the advantage is that all these reactions are happening within this microwell, and that also is advantageous compared to other microfluidic techniques because most of the reactions are happening within this microwell in a more controlled environment. So again, with this, the advantage of the scanner is that from the scanner, we can start looking at these microwells and see the rate of the doublets, or if most of the microwells have a cell. So here, all these gray dots are the beads, and then the colored structures of the cells of the nuclei. So you can see here that there's no doublets here, at least in this picture. And then we can also see that a few of the cells have this, a few of the microwells have the one cell with a microbead. So we can use this to quantify before we go into sequencing this these flow cells. So these come as an 8-lane chip, and the cost of this chip is $10,000. So it translates to around $1,250 per lane. And in each lane, we can capture up to 40,000 cells or nuclei. So just to summarize and compare the stats across these single cell platforms now. So when we think about doing individual sample in 10x genomics, the per reaction, the cost is around $2,000 or $2,500. That translates into 42 cents per cell. And then we need this Chromium 10x instrument. And we have an expected output of 4,000 to 8,000 cells or nuclei. And in our experiments that Bruno and Graham has done along with me, we see that the median UMI per cell is 15 to 20,000. And when we use nuclei, we get 2,000 to 3,000 UMIs per nuclei. So the quality is high, but as Sam was mentioning, there's high potential of clogs, particularly when we use protoplasts. And then there are options for single cell ATAC-seq as well as multi-ohm. And for the scale biosciences, we have to buy the kit for 96 samples that cost $10,000. And it comes down to 8 cents, around 8 cents per cell. We don't need any specific equipment. And because we have the output is 125,000 in total, but if you count 96 samples, it's around 1.3 thousand per sample. And we see half of what we see from 10x genomics, that is, we see 7,000 to 9,000 UMIs per cell and 1,000 to 1,500 UMIs per nuclei. And the advantage, as I mentioned, is that we can fix the samples and then we can mix multiple cells and nuclei from different species because we can multiplex up to 96 samples in this. They have a single cell ATAC-seq, and my lab is also working on the single cell methylome kit. They are very expensive at this point, but still we're thinking that we can get good information from these single cell methylome kits. And with the BD Rhapsody, each lane would cost you $1,250. That translates to 3 cents per cell because we get an output of 30 to 40,000. And we can just do this with this $15,000 instrument, the HT Express. We may also need a scanner, which is around $50,000, but just this HT Express should be sufficient to generate the libraries and sequence them. We saw similar data quality compared to 10x genomics. We see 15 to 20,000 UMIs per cell. We did not test nuclei. So this we think is more reliable. We don't see any clogs, and they have single cell ATAC-seq and single cell multi-ohm kits available. And as I mentioned, we have also used Parse and Fluent. And similar to Sam's experience in our experiments, it did not work well for us. We saw, I think, 90 to 95% of the libraries were all rRNA due to this random hexamers in the cDNA step. So we didn't work further on Parse and Fluent biosciences. But I think with these three technologies, we can see that both Scale and BD Rhapsody are helping us into expanding into different types of experiments. With that, I would like to acknowledge that most of the work that I described here was done in Ken Birnbaum's lab at New York University, where I was a postdoc with Bruno and Graham. And then I would also like to acknowledge my new lab at UC Riverside, where we are trying to explore these techniques. And just to give a shout out to my lab and my contact information for anyone who's interested to learn more about these techniques. And with that, I'll be happy to take any questions.
Thank you very much, Sunil. And again, feel free to post your question via the Q&A session. I have a first questions for you, Sunil. So you show pretty well the level of overlap of the transcriptome between different root cell types, such as the phloem cells, for instance. Did you try to analyze a different organ from the canopy and then conduct similar comparative analysis to fully estimate, even between very different organs, the level of redundancy of those genetic programs?
Yes. So because of time, I could not talk about the project where we are also looking at how different organs in both root and the shoot can have specialization in cell types that leads to distinct functions. So we look at what is conserved across meristems in the shoot and in the ear inflorescence of maize, and then try to figure out how specialization in certain cell types, even within those that are highly conserved like xylem and phloem, we still see specialization that are important for their function in the shoot, for example. So we are working on that, but I did not present about the results from that work.
Okay. Just by curiosity, did you notice also some differences related to the organ of origins? Just again, to think about the contextual information in terms of cell types and cell functions surrounding, for instance, the phloem cells.
So are you asking whether there is any correlation between specialization and the origin of those cell types?
Yeah.
Right? Yeah. So I think even within these root comparison, we see that when we look at lateral roots which emerge from the primary or the seminal roots, they are much more different to their primary root. So for example, the primary lateral has more similarity with seminal lateral root than with its own primary root. So this suggests that there are some examples that suggest that the origin of these cell types, they do have some effect, but more than that, it's the specialization that happens at the organ level that becomes more important than their origin, at least in the root data set.
Thank you. Waiting for more questions, we can very likely open now. Oh, maybe Hervé, you want to ask a question?
No, it's okay. I'll let you follow.
We have one more questions for you, maybe Sunil, before we can really open to a broader Q&A sessions for all three speakers. So it's from Trevor Freeman. Can you talk about how you projected single-cell RNA-seq datasets from different species into a common feature space? So how did you project these single-cell datasets?
Yeah. So for this particular dataset, Bruno used one-to-one orthologs in maize, sorghum, and Citaria, and then used those genes to project them into two-dimensional UMAP plots. So the integration here we did using this one-to-one orthologs, but there are new there are papers now that has come out recently that shows that instead of using one-to-one orthologs, which can be limiting when you are comparing species which are evolutionarily far off, you can also use gene co-expression information. So Michael Pasalacqua from Cold Spring Harbor, he has this paper where they're using bulk RNA-seq co-expression data to get information on these orthologs, because plants have these paralogs, and these paralogs can have differentiation of function. We need to understand which genes are co-expressed, and we can use that information to integrate data set from across species.
Thank you. So yeah, I think we can open to a broader spectrum of questions now for any of the three speakers. Again, feel free to use the Q&A sessions if you have any questions.
I think I'll go ahead and stop the recording now for the, if we're going to the general.
Yep.